Okay, good afternoon, everyone. Thank you for joining us for the Sahyadri Symposium. Before we begin, I'm going to give a brief overview of Plant Postdocs. Plant Postdocs was founded in 2019 by Arif Ashraf and Sunil Kenchan Mane Raju, and they recognized that there was a need for a plant postdoc community. And so this community is intended to help us reach the next step in our careers, whether it's in academia or industry or other elsewhere as well. So the current leadership team consists of myself and Xiaohui. We have two new members, including Becca and Sonal. And we also have Ariadna, Israel, and Hannah. And so if you have any questions about Plant Postdocs, feel free to reach out to us or to contact us through our Plant Postdocs email. There are many ways that you can get involved with Plant Postdocs. You can check us out on our website. There we have a lot of different resources that are intended to provide career development opportunities. We also have an active social media account on X or Twitter and Blue Sky. And you can see all of our past webinars on YouTube. And so this webinar will also be recorded and will be posted following today. And so next, I'm going to pass it over to Sonal, who will be moderating today's webinar and introducing our speakers.
Hi, everybody. I'm Sonal, and I'm here to moderate the third Sahyadri Symposium. So this is going to feature our talks from the awardees of the 2025 round. And a little background on the name is Sahyadri mountain range is the upper and central part of the Western Ghats in India. And as the mountains are magnificent, so are our awardees today. So we are going to talk about their research. They were matched with mentors who helped them through the application phase, the journey, and Plant Postdoc teams were there by their side during this difficult phase. And without further ado, I'm going to talk and introduce Dr. Penelope, Dr. Erika, and Dr. Elena today. And they are all wonderful postdocs, and they have amazing plant research experiences, which they're going to share with us today. So our first speaker of the day is Dr. Penelope Lindsay. She is a research associate from the Liebhoff Lab in Oregon State University, and she's going to talk about clavata receptor networks that shape maize development. So Penelope, can you please enlighten us with your amazing work?
So first of all, I wanted to say thank you for that lovely introduction. It's a great privilege to be nominated and invited by peers to be able to participate in this great opportunity for assistance during the job application process. And so I'm really excited to share my work with you all today, which is about antagonistic interactions between clavata receptors that are important for maize ear development. So maize inflorescence development is controlled by the clavata signaling pathway, which is a pathway that's been quite well described over the years for Arabidopsis. And so at the heart of this signaling pathway, you have a transcription factor called WUSCHL, which is expressed in a subdomain of the meristem. WUSCHL can move intercellularly to induce the expression of small secreted peptides called Cli peptides, which are in turn perceived by a suite of receptors. This includes clavata-1, which in maize we call thick tassel dwarf 1 or TD1, clavata-2, which we call Fe2 in maize. And through the analysis of overproliferating meristem mutants in maize, we actually discovered a novel component of this pathway called Fe3. So you can see on the right here the inflorescence meristems of maize, and in these mutants, the inflorescence meristem is massively overproliferating compared to wild type. As a result of this, we have what we call fasciated ears, which are these disorganized ears with an increase in the number of kernels, especially around the perimeter of the ear. So since we discovered this novel component of the clavata signaling pathway, we wanted to better understand its mode of action in regulating meristem development. It's a leucine-rich receptor-like protein that lacks a kinase domain, which suggests it needs to interact with other kinases to be able to participate in signal transduction. We did crosses between Fei3 and TD1 and Fei2, other known clavata receptors, but we saw an additive genetic interaction between Fei3 and these receptors, indicating that Fei3 likely acts in parallel to these other receptors. Previous work from the Jackson lab showed that Fei3 can perceive the Cli peptide FCP1. We performed these peptide assays where typically when you apply a Cli peptide, you have either a smaller shoot apical meristem or smaller root compared to a scrambled Cli peptide control. But in this Fei3 mutant, we see insensitivity of this response. However, we collaborated with Yoshikatsu Matsubayashi at Nagoya University and found that Fei3 does not actually directly bind FCP1. Interestingly, though, we did see that FCP1 bound to barely any meristem 1, which is another LRR receptor-like kinase that is closely related to clavata1. And so previous work in Arabidopsis has shown that higher order BAM mutants play pleiotropic roles in development. So everything from shorter plants to altered inflorescence development, altered pollen development, root development, and even venation patterning. And as I mentioned before, you need to have higher order mutants to see these strong effects. And there are 3 BAMs in Arabidopsis, but in maize there are 7 BAMs. So I took a closer look at the expression pattern of the different BAMs, and I found that one BAM in particular, BAM1D, had an expression pattern most similar to Fei3. And I generated a native promoter-driven translational fusion of BAM1D with YFP and crossed it into an existing RFP Fei3 line that we had. And I saw that the 2 transgenes were co-expressed. So this led us to hypothesize that perhaps Fei3 and BAM1D form a receptor complex. So to be able to determine whether or not the 2 proteins interacted, I conducted an experiment called proximity labeling with Fei3 and BAM1D. And in this type of technique, we have an engineered biotin ligase called TurboID that we fuse to our proteins of interest. And then upon treatment with biotin, the TurboID will biotinylate any neighboring proteins, and then we can pull out all those biotinylated proteins and do a mass spec analysis to see what's in proximity of our proteins of interest. And so with great help from a visiting master student in the lab, Panagiotis Pumpas, we performed a TurboID affinity purification mass spec experiment where we took these different lines along with a non-transgenic control. We treated shoot apex tissue with biotin and then performed the protein extraction purification with streptavidin to get out all the biotinylated proteins and this mass spec analysis. And from this approach, we saw a significant overlap in the enriched proteins in the Fei3 and BAM1D samples. Some of these proteins included signaling proteins, so RAC1 is a G protein scaffold, as well as proteins related to receptor-mediated endocytosis, such as this EHD1 protein. Additionally, we saw that BAM1D was biotinylated by Fei3, supporting the idea that the 2 proteins interact. So I next wanted to assess the biological relevance of this interaction. So We first tested whether BAM1D could perceive the same peptide as Phe3. And indeed, we saw that BAM1D is insensitive to FCP1, although it does appear insensitive to a broader range of cleaved peptides than Phe3. We also performed the peptide binding assays with BAM1D and found that BAM1D can bind FCP1. So we next wanted to assess BAM1D's role in maize ear development. So I crossed a Phe3 null line that we had in the lab to 3 different BAM1D alleles to assess its role in ear development. And so what I hypothesized is that there would be a synergistic genetic interaction between Phe3 and BAM1D because BAM1D expression is broader than Phe3, which didn't really show in this talk, and higher order BAM mutants have pleiotropic developmental effects. So this combination may unlock a phenotype that you would not see with either of the single mutants. So first I looked at the inflorescence meristem size, and I found that BAM1D single mutants have smaller meristems compared to wild type. But intriguingly, I did not see any change in the meristem size in Phe3 BAM1D double mutants compared to Phe3 singles. This suggests that Phe3 is epistatic to BAM1D in the control of inflorescence meristem size. However, I saw something very interesting in the mature ear phenotype where typically we see this increase in kernel row number, which is the number of kernels that are around the perimeter of the ear of corn in Phe3. But this effect was suppressed in the double mutant compared to the Phe3 single mutant, even though there is no change in kernel row number in the BAM1D single. So this indicates there is a synergistic effect between Phe3 and BAM1D in the control of maize ear size and in particular kernel row number. So what I've told you today is that BAM1D and Phe3 are co-expressed, that they perceive the same cleaved peptide, FCP1, that they interact in meristematic tissue, but that they seem to play antagonistic roles in maize ear development. So what I'm really curious to address in the future is sort of how Phe3 modifies the signaling capacity of BAM1D. It's a receptor-like protein, so my hypothesis is that perhaps it's altering the binding affinity for certain cleaved peptides through this physical modulation of the interaction. So in general, as I mentioned before, Phe3 and BAM1D are partially co-expressed. They're not completely co-expressed. And this work in combination with the greater Clavata literature suggests that perhaps there are many heterogeneous receptor complexes that are perceiving cleaved peptide ligands. And I'm really interested in this paradigm. So this is something that the Elwitz lab has proposed in animal signaling systems at Caltech, where you have several different heterogeneous receptor complexes that are perceiving many different ligands. And this is what really allows a fine-tuned response in development or in the trade-off between development and perception of the environment. And I could certainly see something analogous happening here because while I just highlighted a few of the Clavata receptors, there are a decent number, particularly in maize, but not nearly as many as there are cleaved peptides, where in maize there are 49, for example. So to me, these heterogeneous receptor complexes would kind of allow for this more fine-tuned response to cleaved peptides. So I'll be really excited to explore this question more in the future. So with that, I'd like to thank you all for listening today. I'd like to acknowledge my previous lab, the Jackson Lab, which provided a really awesome environment for doing this work. I want to acknowledge my current lab, the Liebhoff lab, for allowing me to actually continue a lot of my own independent research. And I'd like to acknowledge collaborators at the University of Missouri, at the Sainsbury lab, Iowa State, and the Carnegie Institute. And if you'd like to hear more, or read more, feel free to scan this QR code here. So thank you.
A wonderful talk, Penelope. It was really crisp and exciting to know about your work. So, people in the audience, please feel free to ask questions. We can allow a question or two before we head on to the next speaker. If you're comfortable, you can unmute yourself or go ahead and write it in the chat box, whichever is comfortable. So while we receive questions from our audience, I was just curious about the technique that you use, the proximity ligation. I have seen collaborators try working on it. So did you face any issues with non-specific binding during your studies of the pull down, or was it pretty clear for you?
Yeah, so I think certainly with this technique, you will encounter some non-specific biotinylation. And in fact, people will often try to use a compartment-specific turbo ID molecule, or BAIT as the control. However, in my experience, I tried this initially, and I think I became very unlucky with my choice of negative control, because it was one of the most strongly biotinylated proteins by this control was actually BAM1D. So I think I would just say this is maybe a precautionary tale, and being very thoughtful about the choice of negative control for these types of experiments, because they can play a really big role in terms of what you see.
Okay. Thank you. And just one quick question: how long was your protein in general? Like, because LRRs, these have a very long domain. Which, like, was it too big for you, or was the protein domain specific here?
Yeah, so the BAM1D protein is about 1,000 amino acids or so, so pretty big. And Phe3, and it has about 15 LORs or so, and Phe3 has fewer, probably in the 8 to 10 region. Yeah, so it's a smaller. Yeah.
Okay. So you use the full thing? Wow, then it's amazing, because I see collaborators struggling with the expression with such big and large proteins, and then the whole experimental assay. But thank you so much for the advice. So moving on to our next speaker today is Dr. Elena Perry, postdoctoral fellow in the Environmental Genomics and Systems Biology Division. Yeah.
There might have been a question. I think Song Liang?
Yes.
Oh, yeah, sure.
Yeah, there's still a few minutes for me to ask questions for the Penelope. Okay, and that's a wonderful talk, and thank you for sharing your work. My question is, you mentioned that the Phe3 cannot bind FCP1, but instead the BAM1D binds FCP1. So my question is, do they need to work together to bind it, or BAM1D itself can have a very high binding affinity?
Yeah, so BAM1D is able to bind FCP1 by itself, but definitely something I want to do in the future is understand whether Phe3 can modulate the binding affinity for FCP1. So I want to do some ITC experiments to be able to assess that, but I haven't done them yet. But I think it's a super interesting question. Yeah.
Is it because the LRR domain in the FCP in Phe3 shorter than BAM1D or something else?
So it's not just that it's shorter. I actually just did a very fine scale analysis of the amino acids in the Phe3 LRR domain, and it actually lacks a lot of the amino acids that are typically associated with the Cli peptide binding. So yeah, it's interesting. And I wonder about the evolutionary aspect of that, but that again will be another future avenue of study, I think. Yeah.
Yeah, thank you. Thank you. That's my question. Thank you.
Thanks.
Yeah. Okay, sorry for that, by the way. If there's anyone else, please feel free to unmute. Okay, then. So this time again, moving on to our next speaker, Dr. Elena Perry. She is a postdoctoral fellow in the Environmental Genomics and Systems Biology Lab division of at Lawrence Berkeley National Laboratory, where she's studying the native methane-eating bacteria and their influence on the structure and function of rice microbiome. So today her talk is entitled Methane Supports Diverse Microbial Communities Enriched from Field-Grown Rice. So over to you, Elena.
Thank you so much for that introduction, Sonal, and to the plant postdoc community for this opportunity to share some of the work from my current postdoc. Today, as Sonal mentioned, I'm going to share a short story about my current work on how methane can support diverse microbial communities in field-grown rice. Sorry. So all plants are colonized by billions or even trillions of bacteria, which can be found in all parts of the plant, from the roots to the stem and leaves, and even the seeds. Together with other microbes such as fungi, these plant-associated bacteria make up a microbial community that's known as the plant microbiome, which differs across different types of plants as well as plants grown in different environments. These plant-associated bacteria can affect host health as well as ecosystem health in a variety of ways. For example, some bacteria are capable of fixing nitrogen, which means that they can convert atmospheric nitrogen into a form that is bioavailable and can be used to promote plant growth. For example, in this photo, these are showing root nodules, which are found on a type of plant called legumes. This is a specialized organ that's dedicated to housing nitrogen-fixing bacteria to benefit the plant. Other bacteria can either cause or prevent various plant diseases. Some bacteria associated with plants can also solubilize various minerals that are required for plant growth. And other bacteria can either produce or consume greenhouse gases that affect climate change, such as methane or nitrous oxide. And this is relevant to the work I'm going to present today. Specifically, in my current postdoc, I've been studying microbial interactions in the rice microbiome. Rice is typically grown under flooded conditions in a type of field called a paddy. And this is historically done in order to suppress weed growth because rice is very water tolerant, unlike most weeds. However, this also leads to the production of significant quantities of methane through the microbial anaerobic decomposition of organic matter. This methane can then either bubble up or diffuse up into the atmosphere. It can also be transported through the plant vasculature itself and again escape to the atmosphere. However, some of the methane is actually consumed by another special type of microbe known as a methanotroph. These are bacteria that can literally eat methane, using it as their carbon and energy source. And through a series of reactions that require oxygen, it ultimately converts the methane all the way to carbon dioxide, which then gets released. These methanotrophs, because they consume methane, have the potential to further improve the sustainability of rice cultivation if we could increase their activity in the rice fields. However, not much is known about what the other potential consequences of this could be. Some prior work has suggested that methanotrophs can indirectly affect various aspects of nitrogen cycling in rice paddies. But exactly how this is happening at a mechanistic level and how we might be able to alter that is not well understood. So to be able to address this, we decided to first focus on getting a better understanding of which bacteria in the rice microbiome can potentially benefit from methanotroph activity. To pursue this question, we took a very old microbiology technique called enrichment culturing. Specifically, we took different parts of field-grown rice plants, including the stem, the roots, as well as the rhizosphere, which is the 1 to 2 millimeters of soil that's very tightly adhered to the roots. And we diluted these into a very basic mineral medium, which only contains some vitamins, some basic mineral salts that are required for growth, as well as a nitrogen source in the form of nitrate, but no exogenous carbon source except for methane, which we provided in the headspace atmosphere of these cultures. The idea was that by providing only methane as the carbon source, these cultures should enrich for the bacteria that are able to oxidize or eat methane, as well as any of their partners from the rice microbiome. We grew these cultures for 12 weeks, and every 4 weeks we diluted them at a 1:10 ratio to further dilute out any members of the community that are not involved in the cycling of methane. Additionally, at each time point, we profiled the community composition using a technique known as 16S rRNA gene amplicon sequencing. This is a very useful technique in microbiology because the 16S gene exhibits a lot of variability across different taxa in a way that's correlated with phylogeny, and therefore allows us to use this type of sequencing to get a compositional analysis. What we saw was that by the final time point of these enrichments, the communities were still remarkably diverse. What I'm showing here is essentially a stacked bar plot where each column represents one of the enrichment cultures, which is coming from different types of samples from different fields, either Biggs Field or Staten Island or SI, which are two different locations in California. Different parts of the plant, including the rhizosphere, root, and stem, as I mentioned, as well as 3 different dilutions for each of those original samples at the outset of the experiment. And then within each of these stacked columns, each different color represents a different taxonomic group. And I've highlighted with different colors the major taxonomic groups that were enriched to more than 10% final relative abundance in at least 2 of the enrichments. And then further within each of those colors, the white lines are separating different specific sequences, which putatively represent different species within those taxonomic groups. And overall, this represents their relative abundance to each other within each community. What was very interesting was that only these yellow and tan taxonomic groups are known methanotrophs that can eat methane. And while they did reach a high relative abundance in many of the enrichment cultures, as we would expect, there was also many other taxa that were enriched to quite high abundances and frequently made up a majority of these communities by the final time point. And this is quite surprising in some ways because these other taxa cannot eat methane. What was also interesting is that the majority of these taxa that we detected have not previously been identified as being related to methane cycling in rice paddies using other techniques, suggesting that this enrichment culturing technique might be a more sensitive method for figuring out which bacteria interact either directly or indirectly with methanotrophs in the rice microbiome. However, the fact that so many of these taxa are not known to be able to eat methane raised the question of how is it that methane-eating bacteria are supporting the growth of all these other community members under methane. To begin to dissect this mechanistically, we decided to generate a collection of bacterial isolates from these enrichment cultures. And we did this through a multi-step process where we serially diluted the cultures and then plated them onto different solid media containing different carbon sources, including methane to select for the methanotrophs, methanol because that's the immediate downstream oxidation product of methane, and it's known that some bacteria can eat methanol but can't eat methane. And then we also use the rich medium with a broad range of carbon sources to select for as many other members of these communities as possible. We then picked individual bacterial colonies and streaked them out several times to obtain pure isolates that have just one taxon. And in total, we obtained 65 isolates that represent 22 different taxonomic families, and 3 of these are methanotrophs. And overall, we got a pretty high representation of those taxonomic groups that I mentioned earlier were particularly enriched in this set of enrichment cultures from the rice, including 13 out of 18 of those groups. When I performed genomic analysis for 40 of these isolates, I found that consistent with the prior suggestion that methanotrophs may be able to indirectly affect nitrogen fixation in rice paddies, many of these isolates possess the NIF genes that are required for nitrogen fixation, suggesting that they could potentially benefit the plant under nitrogen-limited environments. On the other hand, many isolates also contain various steps of the denitrification pathway, which converts bioavailable nitrate all the way back to atmospheric nitrogen, causing nitrogen loss from agricultural fields, which is not desirable. So in the future, we hope to be able to use this diverse set of isolates to gain a better mechanistic understanding of how it is that methanotrophs can influence both ends of the nitrogen cycle in ways that are both positive and negative for the plant. To begin to mechanistically dissect how the methanotrophs are supporting the growth of these different bacteria, I turned to a functional genomics technique called random barcode transposon site sequencing, or RBT-seq, which was invented in the lab where I'm currently doing my postdoc. This technique uses mobile segments of DNA called transposons to randomly insert into and disrupt the function of various genes throughout the bacterial genome. And what's unique about this technique is that each copy of the transposon that we use has a unique short segment of DNA called a barcode. And what we can do then through high throughput sequencing is create an association between which barcoded transposon has leaped into which part of the genome, and therefore associate, barcode 1 with gene A, barcode 2 with gene B, etc. What this then allows us to do is to pool all of these mutants together, up to hundreds of thousands at once, and grow them under different experimental conditions. And again, using sequencing, this time of just the barcode sequence, we can track the relative abundance of these mutants over the course of the experiment. And by comparing the relative abundance of each barcode at the end of the experiment compared to the beginning, we can use those changes in abundance to calculate a fitness score for each of the genes in the genome, meaning how important are they for growth in that particular condition. For example, in the case of barcode 1, we can see that it increased in the course of the experiment in relative abundance, meaning that this mutation actually was beneficial for the bacterial growth, but what that really means is the gene that was mutated probably is detrimental. Conversely, you can see in other cases in this toy example that the barcodes dropped out of the population, meaning that the genes in which they were inserted, creating a loss of function mutation, are actually very important for growth under this condition. And there's also usually a large number of genes in the genome which do not have a direct fitness effect under any given condition, and therefore their relative abundance remains unchanged, as in the example I'm showing here with barcodes 4 and 6. And taken together, this technique allows us to very rapidly and economically screen the function of every gene in the genome for different bacteria under a very large number of conditions. I decided to apply this technique to dissecting the question of what the non-methanotrophs are eating in methane-fed communities. And specifically for proof of concept, I focused on one pair of bacteria, Methylocyanus, which is a methanotroph, and Pseudomonas, which was one of the isolates. Both of these actually were isolated from our rice enrichment communities. What you can see here is that during growth on methane, Pseudomonas itself is not able to grow because it can't eat methane. However, when we grew it in the presence of the methanotroph, it grew to a very high abundance, suggesting that it is successfully eating something from this methanotroph. And to show that it isn't simply killing and directly eating the cells of the methanotroph, I also grew it in the presence of what we call spent media, where I first grew the methanotroph by itself under methane, and then I spun down the media to remove the cells. I also filtered it, and then I fed that to the Pseudomonas. And Pseudomonas was still able to reach a pretty high cell density, indicating it's just growing on some excreted products that the methanotroph had released into the medium. When I then used RBT-seq on the Pseudomonas, I was able to find a number of genes which were specifically important for growth in the presence of the methanotroph, but not in our control condition. And what we noticed was that the top 3 genes that were specifically important in the co-culture are all known to be required for acetate catabolism, suggesting that Pseudomonas is most likely eating acetate that was excreted by Methylocyanus. Finally, while this is a very good proof of concept for the ability of our functional genomics technique to dissect interspecies interactions in these methane-fed communities, this is just one pairwise example. And the original enrichment communities, as well as the overall rice microbiome itself, is much more complex and has many more taxa that are interacting with each other. So to lay a foundation for dissecting interactions in this more complex context, I decided to see if we could recapitulate the dynamics of the original enrichments with a more simplified set of the communities, focusing just on the species that we had isolated. So what I did is I took a set of enrichments, for which I'm showing 6 different ones here, for which we had pretty good representation among our isolate collection of a high proportion of the original diversity in those communities. I then combined those isolates in the relevant combinations and grew them across 3 biological replicates for 2 weeks with 1 passaging step after the first week. And what you can hopefully appreciate here in these plots of relative abundance is that the overall dynamics and community structure of these, what we call synthetic communities, constructed of the isolates, was quite similar to the original enrichment communities, suggesting that we now have a tool that allows us to recapitulate a fairly relevant degree of diversity and complexity using isolates where we can control the input abundance as well as perform genetic manipulations on these bacteria to discover how they interact with each other. And what I've shown here with the arrows is just pointing out all the different taxa from these communities for which we now have RBT and seq libraries, which will allow us to do those mechanistic experiments in the future. So to summarize what I've told you today, methane, which is highly produced in flooded rice paddies, can directly and indirectly support diverse members of the rice microbiome. And I've also shown a proof of concept for how we can use high throughput functional genomics to mechanistically dissect bacterial interspecies interactions in plant microbiomes. Going forward, I am planning to combine this RBT and seq technique with our synthetic communities to more comprehensively map methane-driven food webs in the rice microbiome beyond the pairwise example that I've already shown. And also to use this RBT and seq technique to uncover the molecular mechanisms of rice colonization by these diverse members of the rice microbiome. Ultimately, by combining the resulting insights from both of these approaches, we hope to be able to engineer the rice microbiome to maximize its beneficial effects like nitrogen fixation or methane consumption and minimize unwanted effects such as denitrification and nitrogen loss. And with that, I would like to thank everyone who has helped me with this project, especially the PI who initiated the rice enrichments, Dawn Schinicki, as well as Adam Deutschbauer, who's the head of the laboratory, as well as my funding sources and Berkeley Lab. And I'll be happy to take any questions.
Amazing talk, Elena. There's one quick question that we can address. And Zahur is asking, does these microbes in nature and in lab conditions behave differently as in natural conditions? Are there more ecological interactions happening? Could you answer her question quickly?
Yeah, this is a very good question. For certain, the conditions that we use in the lab would affect how they potentially interact and behave. And so it's important for us to use our knowledge as much as possible of the natural environmental conditions to stimulate that as best we can. And so I'm planning to do this at different levels of complexity. For example, we can modulate oxygen concentrations, which we know are quite relevant in these environments. We can also modulate which nitrogen source we provide, which may be relevant. And ultimately, we can also scale up to put these microbes back on the plant, but in a more controlled setting in the lab, and then, eventually work our way back to the original complexity of the field setting and see if our observations still hold true. And as for the question of, are there more ecological interactions happening than molecular ones? I guess I would argue, like, sort of the thesis of my approach is that ecological interactions, especially for microbes, are ultimately underpinned by molecular interactions. And so it is, in fact, the exchange of different metabolites or, interactions of different proteins from different bacteria or other molecular components that results in the ecological dynamics, if that makes sense.
Yeah. Thank you, Elena. So our final speaker of the day is Dr. Erica Lawrence Paul. She is a postdoctoral scholar at the Pennsylvania State University, working with Jesse Lasky, and her research examines as to how developmental transitions shape plant responses to environment and contribute to a local adaptation. The title of her presentation is on screen, talking about the importance of developmental context and state-specific strategies for drought-resilient plants. So thanks, Erica.
Thanks, everyone. Yeah, so today I want to think about how plants, as they're going through different developmental transitions, might interact differently with their environment and have different responses to stress. Okay, so as plants transition through different phases of development, they have changes in morphology and physiology that we are hypothesizing will also impact their stress tolerance. So here in this figure, which we should go back here, we see that different colors are representing stress tolerance, with blue indicating low stress tolerance and red indicating high stress tolerance. We're also hypothesizing that different genotypes could have different patterns of stress tolerance that shift as they're moving through these different developmental transitions. So we also have represented here two different genotypes, A and B, that you can see have different patterns of stress tolerance indicated by these changes in color. So we know that different environments have different patterns of environmental stress. So here on the bottom, we're showing two representative environments. Environment 1, where environmental stress is increasing over the growing season or increasing over time. And environment 2, where we see that there's a high period of stress earlier in the growing season or earlier in time, followed by periods of lower stress. And so these differences in environments and these differences in phase-specific traits and phase-specific stress responses can contribute to environmental adaptation. So for example, here we would see that genotype A is probably best suited for a place like environment 1, where we have some alignment of those stress-tolerant phases of development with when the plant is most likely to experience environmental stress. And a genotype like genotype B would be better suited for a place like environment 2, again, aligning those stress-tolerant phases of development with that period of high environmental stress. So one of the transitions that happen in all plants is between the juvenile and the adult vegetative phases of growth. This is known as vegetative phase change. So this happens prior to reproduction, and it's a phase of transition that's ubiquitous in all plants, but is pretty understudied. And so this transition is actually the focus of my research and what I'm going to talk to you about today. So I wanted to ask a few different questions surrounding vegetative phase change and how this transition shifts stress response and sort of the mechanisms underlying those shifts in stress response. So the first question here is adaptation to drought stage specific? So do we actually see differences in stress response between the juvenile and adult vegetative phases of growth? Are drought response genes stage specific? So do we see any differences in the genes that are underlying drought response in these two different phases? And then how does natural variation in vegetative phase change contribute to drought adaptation? So what kind of natural variation in the timing of this transition do we see? And is it at all correlated with the environment that these plants are from? So to do this, I used 158 genotypes of Arabidopsis thaliana. So these are mostly genotypes from Europe and Asia. And so you can see here, the first thing I did was look at when do these plants transition. So here we're looking at the first adult leaf. So this is the leaf position where the plants transition to the adult phase. We had really nice variation with some genotypes transitioning as early as leaf 4, others not transitioning until leaf 18. And so this was a span of about 16 days, which in a plant with a short life cycle like Arabidopsis is a pretty significant period of time. So these plants were grown in 3 different conditions. The first one is a well-watered control. So the plants were well-watered throughout their entire lifespan. We have a juvenile drought treatment. So these plants experienced a 10-day drought during the juvenile phase of development. They were then moved back to well-watered conditions to finish out their life cycle. And the last one is the adult drought treatment, where plants experienced a 10-day drought during the adult phase of development, and then again were moved back to well-watered conditions to carry out the rest of their life cycle. So basically the difference there is they're experiencing a 10-day drought. One is during the juvenile phase and one is during the adult phase. So here we saw that the developmental phase that a plant experiences drought does have an impact on those fitness responses to drought. So we're looking at the number of fruits that were produced by plants of all these genotypes in our 3 different treatments. We can see that both juvenile and adult drought plants produced fewer fruits than the well-watered control, but that the majority of our genotypes produced even fewer fruits when they experienced that drought in the juvenile phase compared to if they experienced the drought in the adult phase. And the super interesting thing here was that we had really significant genotype by environment interactions. So this is revealing that there is stage-specific drought tolerance. So it wasn't just that one genotype was drought tolerant across the board and other genotypes were drought sensitive. It was really specific to when the plants actually experienced that drought. And so just to kind of highlight what I'm talking about here, chosen 3 different genotypes that show different patterns of phase-specific tolerance. So this first one, OV0, had one of the highest fitnesses in that adult drought treatment, produced some of the greatest numbers of fruits, but was kind of in the lower half of our genotypes when it experienced a drought in the juvenile phase. So you can see here, very big difference in the number of fruits produced in these treatments. This next genotype, CROT0, had pretty stable response to drought. So it produced a very similar number of fruits in both our juvenile and adult drought treatments. However, because of all of the other shifts in genotypes, that rank order changing really shifted. So while it had some of the highest fitness in that juvenile drought treatment, it was kind of just in the middle of the pack for the adult drought treatment. And then lastly, this AA0 genotype, kind of towards the top third in terms of fitness for juvenile drought, but one of the lowest fitness genotypes for adult drought. And so the reason why this is really interesting and important is because as we move forward and we're trying to identify traits and genotypes that are best suited for drought conditions, if we're only focusing on one time point or one developmental phase, in this case, we might have chosen OV0 and used that genotype in our drought conditions. But if this plant is in an environment that experiences drought earlier in development or earlier in the growing season, it's really not going to be successful because that juvenile drought shows us that it's very sensitive to drought in that phase. So really considering development and the fact that development can interact with responses is really important. So here we also looked at a couple of different traits. So this is rosette growth during that 10-day period when plants were experiencing drought. So here we have during the juvenile phase, that 10-day period, how much that rosette grew for plants that were well watered in that phase or experienced a drought in that phase. And we can see a significant reduction in rosette growth for these juvenile drought exposed plants. However, if we look during the adult phase and we see our plants in the adult phase that were well watered or experienced a drought, we see no significant difference. So again, indicating that these drought responses are phase specific and that we are seeing, it is important to see whether, the drought is being experienced in the juvenile or adult phase. And then our last trait here is water use efficiency. So we used carbon isotopes to look at the ratio of carbon gained per water lost through the stomata. So here, these higher values are higher water use efficiency, meaning that the plant lost less water as it was photosynthesizing. And then these lower values are lower water use efficiency, where the plant used more water. And so here, again, we're looking at tissues that were produced during that time period where juvenile drought plants were experiencing a drought, either for plants that were well watered during that time period or plants that were in that drought treatment. We see no significant difference in water use efficiency here in the juvenile phase. But if we look in the adult phase, we do see a shift in those drought exposed plants to a higher water use efficiency. So again, just another trait indicating that these plants are responding to drought differently depending on what phase they're in. And so because we used this diversity panel, we were able to do a genome-wide association study to look at candidate genes that might have phase-specific roles in drought response. And so here, we used a number of knockout lines for candidate genes that came up in our GWAS to look at the number of fruits that were produced when these plants were grown, either in the juvenile drought in purple or adult drought in orange treatments. So this first group here, this is the Columbia wild type, and the rest of them are different genes, knockout lines. And so we can see here a number of genes showing phase-specific response or phase-specific regulation of drought response in terms of fruits produced. So all of these genes here had an effect during the juvenile phase drought response, but no effect in the adult phase of drought response. We have another gene here that showed the opposite. So this impacted fitness in the adult phase, but not in the juvenile phase. And then a number of lines that showed some sort of interaction with drought response in both phases. But even within these genes, you can still see that there's some differences between the juvenile and adult drought treatments. So still probably some phase-specific roles for these genes here. And then the last thing I wanted to look at was thinking about the timing of phase change, whether that might be contributing to local adaptation and drought adaptation in different environments. So if we look kind of at a global scale at all of our genotypes, we don't see any strong relationships between the timing of phase change and climate, although a lot of these areas are vastly different in climate, and so are going to have different climate pressures that are going to be selecting on different developmental transitions. So when we look kind of at a smaller local scale, we could do that kind of in two groups here where we had a lot of genotypes. So on the bottom here, this is Iberia. On the top, we have Germany. What we're looking at here is the first adult leaf on the y-axis, and then two traits, mean spring temperature and mean fall VPD, or vapor pressure deficit. So vapor pressure deficit, if it's higher, is indicative of a more drought condition. And we are looking at spring and fall because that is when Arabidopsis is naturally growing in the environment. And so we see for both of these regions that genotypes from areas where the mean spring temperature is higher tend to transition earlier. So they move out of that juvenile phase into the adult phase faster. Maybe something to do with drought, or also just, independent responses to temperature. And then when we look at VPD, we see no relationship in Germany. However, we do see a significant relationship in Iberia, where these genotypes from areas with high vapor pressure deficit or higher drought-like conditions are again moving out of that juvenile phase faster, moving into the maybe more drought-tolerant adult phase. And one thing to note here is these values in Germany are actually really low, right? So all of these data points would actually fit in this first part of the graph. So this is just to highlight again, these different areas are going to have really different pressures, environmental pressures on different traits like the timing of phase change. So it's not really surprising that we might see selection from different traits on this in different regions. So just to summarize, we saw that developmental stage in terms of juvenile and adult phases is important to consider when studying stress tolerance. We found fitness, growth, and physiological differences in response to drought, depending on whether those plants were in the juvenile or adult phase when they experienced drought. We did see that the juvenile phase on average is more sensitive to drought, but there was that really interesting genotype by environment interaction in that response that should always be kind of kept in mind and thinking about trade-offs between those two phases. And then we saw that drought response genes can be phase specific, where some genes are important in one phase and maybe not as important for drought response in another. And then lastly, natural variation in vegetative phase change likely plays a role in local adaptation, as we saw some significant relationships between the timing of the transition and these different environmental factors. So I'd just like to conclude by thanking my co-authors and the Lasky Lab at Penn State and funding from NSF and NIH, and thank all of you for your time and attention.
Beautiful talk, pretty crisp. So are there any questions? Okay, so Amy has a question, and she thinks this is cool research and a very good talk. So how different are the genes that respond to drought in adult drought versus juvenile drought?
Yes, you mean what is there, like, are there different mechanisms that are at play? Yeah, I'm not sure. I haven't done too much more than kind of identifying these candidate genes from GWAS, and a lot of what I chose for candidate genes have some known role in drought response or osmotic stress or abiotic response previously described. But in terms of like specific pathways or mechanisms, I haven't done enough to kind of dig in there, but I think it's very likely, because we're seeing differences in the traits that are responding, it's very likely that there's some patterns to the mechanisms that these genes are underlying.
Yeah. And the other part of the question is, have you tried expressing juvenile response genes in adult stages or vice versa?
No, no. So yeah, that would also be interesting. Like if we alter the expression patterns of these genes, what phenotypes do we see in terms of drought response? Yeah, that would be interesting.
Okay. Anyone from the audience? While they prepare the question or something, I had one quick question. You did show some phenotype where the leaf and seeds were different and like during the drought stages. So did you see any difference in the flowering time of these? Because they are directly translated in a way.
Yeah. So there's differences in flowering. I mean, there's natural variation in flowering time among these genotypes, but there's also differences in the plasticity of flowering in response to drought. And that also depended on whether the plants experienced that drought in the juvenile or adult phase. So yeah, there was really cool and interesting results there as well.
Yes. Amazing, amazing. So if we do not have any questions any further, I would like to thank all our awardees and all our audience. Hope you had a great time here. And for concluding the sessions, I'll just pass it on to Amy.
Can everyone see that? So thank you to all of our speakers for some really amazing research talks. We have a few announcements. If you haven't heard, we have two databases that we are putting together as plant postdocs. One is a potential plant reviewer database, and one is a potential plant speaker database, where you will be contacted once you're added to either review a paper or maybe be a speaker. And we're trying to distribute these databases around so that early career people have more speaking or reviewing opportunities. The next announcement is that we are starting a GoFundMe for this year. The funds for this GoFundMe will help support our activities. So in the past, they've been used to either provide an honorarium or a plaque to Sahyadri awardees. But we also have some other expenses that we're hoping to use as funds for. So if you are able to donate or able to share this GoFundMe, we'd appreciate it. And finally, there are many ways to contact us or to get involved. And so we have our social media accounts and our email, and we will follow up on this webinar with a post-webinar survey. So thank you for your time and attention, and we'll see you at the next webinar.